Hi, I'm Mike Dustin from the Skirball Institute and New York University School of Medicine. Today, I'll be taking you through the procedure of forming supported planar bilayers for study of the immunological synapse. So, let's get started.
To make the lipid bilayer, we prepare a solution of 20% nickel chelating lipid and 80% phosphatidylcholine in the appropriate amounts in a glass tube with 1 to 2 milliliters of chloroform methanol. The first step is to evaporate the solvent under a stream of nitrogen gas in a warm 30 to 37 degrees Celsius water bath. This usually takes around 15 minutes. Then, remove any remaining solvent under high vacuum using a lyophilizer for 90 minutes. Next, dissolve the lipids in 2% N-octyl glucoside detergent in tris saline buffer to a final concentration of 0.4 millimolar. This creates a solution of mixed detergent phospholipid micelles. To form liposomes, you dialyze this solution against 3 changes of tris saline to remove detergent and form liposomes. We usually work with volumes on the order of 1 milliliter with 6 millimeter diameter tubing with a cutoff around 10 kilodaltons, and are careful to exclude any air bubbles while clamping the tubing. Sterile filter this solution and do the dialysis under clean conditions, and handle it using 70% ethanol sterilized gloves. This solution should be crystal clear and is stored under argon gas to prevent oxidation. If the solution is turbid, then you have generated multi-walled vesicles and need to start again. In order to set up the experiment, we need to first determine how much ICAM1 and MHC are deposited on a given surface area of nickel chelating bilayer at a given concentration of soluble protein. To do this, we deposit bilayers on 5 micrometer diameter glass beads, incubate the glass beads with protein solutions under conditions that approximate those in the flow cells used for imaging, and then read out the density of bound protein by a flow immunofluorometric assay. FITC-labeled antibodies with known numbers of fluoroscein per molecule are used to detect the surface-bound proteins, and Fluoroscein standard beads are used to calibrate the flow microfluorimeter. We will establish conditions that approximate those on antigen-presenting cells with 200 molecules per square micrometer of ICAM1 and 0.2 to 20 molecules per square micrometer of IEK MCC 91103 complex. Planar bilayers form spontaneously when the liposomes fuse to glass, but only if it is properly cleaned. To clean glass, we use acid piranha solution, a freshly prepared mixture of sulfuric acid and 30% hydrogen peroxide, which destroys all organic material and also leaves the glass surface very hydrophilic. When working with piranha, we wear full protective clothing, including an acid apron and heavy acid-resistant gauntlets. The waste needs to be carefully segregated from organic waste and disposed of properly. To prepare the acid piranha solution, add 75 milliliters of concentrated sulfuric acid to a dry beaker, and then carefully add 25 milliliters of 30% hydrogen peroxide. The solution rapidly heats up to greater than 100 degrees Celsius. Then, immerse the dry cover slips in the solution for 15 minutes. Following this incubation in piranha, dunk the cover slips in purified water, and then rinse them for a minute on each side. Then dry the cover slips using a vacuum source to drain off the purified water. Allow the piranha solution to cool and add to the piranha waste container, which is left with the cap loose, well marked in the fume hood. To form the bilayers, our lab uses Bioptec's FCS2 chambers, which have the advantages of integrated heating. The FCS2 system is on a stainless steel base. that clamps together a microfluidic manifold with a 0.5 mm top gasket, a microaqueduct slide coated with indium tin oxide for heating on one surface and with a fluidic groove on the other side, a dust-free 0.25 mm gasket that defines the height of the flow chamber, and the Piranha-cleaned 40 mm round cover slip on which the bilayer is formed. To assemble the flow cell and form planar bilayers, place the manifold with the tubes directed up on a flat surface. Then place the 0.5 mm gasket with holes positioned over the fluidic tubes and make sure this is seated flat. Gently place the microaqueduct slide with the fluidic channels facing up and make sure it seats flat without applying any downward pressure until verifying that the holes in the slide align with the microfluidic tubes. Then lay the dust-free 0.25 mm gasket with a rectangular cutout on top of the microaqueduct slide so that the channel is lined up with the fluidic channels and there are no major air spaces. Place as many as 5 1 microliter drops of liposome suspension on the surface of the microaqueduct slide in a pattern such that the center-to-center distance between each drop is 2.5 millimeters. The 5 liposome drops may have different compositions, but in this case we will only form 2 bilayers, one with no nickel chelating lipids and one with 10% nickel chelating lipids. Once these drops are in place, carefully place the cover slip down on the surface in one motion. Clamp onto the base as quickly as possible. The liposome drops should make contact with the cover slip. This contact should not be broken since the bilayers have probably already formed, and any disruptions may result in defective bilayers. At this point, it is important to remember to mark the positions of the bilayers with a few small ink spots on the slide to aid positioning of the cover slip on the microscope. Wait 20 minutes to make sure the system has equilibrated. Transfer all the solutions using a 20 milliliter syringe connected to about 20 centimeters of flexible tubing and a 3-way stopcock. Another port of the stopcock is then connected to the flow cell by a short length of tubing to minimize the dead volume between the stopcock and the flow cell. Fill the syringe with Heepe's buffered saline containing magnesium chloride, calcium chloride, glucose, and human serum albumin. Prime the tubing and stopcock so there are no air bubbles. Then connect this to the flow cells and push through 5 milliliters in one motion while watching to make sure no air bubbles pass over the bilayer. Now you will have your bilayers. To introduce the His-tagged proteins into the bilayer, it must first be blocked with casein and charged with nickel. Fill a 1 milliliter syringe with the casein nickel ion solution and connect this to the side port without trapping any air bubbles. After injecting the solution, wait 20 minutes to allow for complete blocking. Then wash the blocking solution out with HBS HSA from the 20 milliliter syringe. Now inject 0.5 milliliters of HBS HSA solution containing the poly-His-tagged ICAM1 and IEK. In this experiment, the ICAM1 is labeled with Psi5, so the LFA1 ICAM1 interactions can be followed by imaging the ICAM1 rearrangement in the bilayer. Allow 30 minutes for the protein to bind the nickel-charged bilayer. While the protein is adhering to the bilayer, secure the flow cell to the inverted microscope using a custom stage insert. Then connect the heating electronics to allow the system to equilibrate at 37 degrees Celsius. Use the marks made earlier to position the flow cell so that the objective is aligned with one of the bilayers, and use interference reflection microscopy to locate the focal plane by focusing on the reflected light image of the field diaphragm. Finally, wash out the unbound ICAM1 from the flow cells. Acquire fluorescence images of the bilayer in the ICAM1 channel in TIRF-M and wide-field modes. This is to confirm ICAM1 binding on the nickel-coated bilayer relative to the nickel-free bilayer. The flow cell is now ready to receive cells. For this demonstration, we will use AND T-cell receptor transgenic T-cells that have been transduced with a retrovirus encoding GFP ZAP70. Wash the cells once with HBs-HSA and then incubate the cells with an Alexa 568 labeled Fab to the T-cell receptor so that we can track the TCR microclusters. After 15 minutes, dilute the cells 1 to 10. This dilutes the H57 antibody enough so that it doesn't interfere with TIRF-M imaging, but maintains saturation of the T-cell receptor as the initially bound Fab starts to dissociate. Then inject them into the flow cell. We image the immunological synapse in 3-color TIRF-M mode. This includes the formation of the LFA1 ICAM1 interaction, the formation of TCR clusters and the C-SMAC, and the sustained formation of TCR microclusters, which recruit GFP ZAP70. Some of these results are obvious during acquisition, whereas others will require post-processing analysis to fully appreciate.
Okay, we've just shown you how to use supported planar bilayers to study the immunological synapse. It's very important for these experiments to work that the T cells are in very good shape, and the HEPs buffered saline solution is not very good for long-term T cell culture, so you want to take the cells out of culture, put them into this media, and then use them very quickly. If you find that you want to do longer-term experiments, there's some very good serum-free media formulations that you can substitute for the HEPs buffered saline that'll keep the cells alive much longer if you want to do longer endpoints such as cytokine production or proliferation of the T cells. So that's it. Thanks for watching, and good luck with your experiments.